You're listening to Medscape's In Discussion series on psoriasis, a podcast where thought leaders and clinical experts share their diverse insights and practical ideas for optimizing patient care. This series is hosted by Dr. Stephen R. Feldman, editor of the Journal of Dermatological Treatment and professor of dermatology at the Wake Forest University School of Medicine in Winston-Salem, North Carolina. Relevant disclosures can be found with the episode show notes on medscape.com or the Medscape app. The topics and discussions are planned, produced, and reviewed independently of advertisers. This podcast is intended only for U.S. healthcare professionals.
Hi, I'm Dr. Steve Feldman. Welcome to Medscape's In Discussion series on psoriasis. This is our fourth episode of our fourth season. In past episodes, we've discussed some of the biologic treatments that have revolutionized the management of psoriasis and other inflammatory diseases. And this has dramatically improved our patients' lives. Those biologics have been around long enough now to have lost patent protection, at least some of them have, but there are no generic versions. Instead, we have biosimilars. In this episode, we'll explore biosimilars with an author of dozens of key papers on biosimilar safety, effectiveness, and economic benefits. We'll discuss what biosimilars are and how they might benefit our patients and the healthcare system. Our guest is professor of medicine and the Peterson Chair in Rheumatology at the University of Massachusetts Medical School, where he also directs clinical research in the Division of Rheumatology. It is my great pleasure to present one of the most wonderful physicians I know, Dr. Jonathan Kay. John, welcome to In Discussion.
Thanks very much, Steve. You're too kind.
Listen, I was looking at your background, and you've been in rheumatology for quite a while, but I noticed some of your first papers were on Lyme disease. Were you anticipating becoming a Lyme disease expert early on?
When I was a senior resident doing a rotation in pediatric rheumatology at Children's Hospital of Philadelphia, there was a patient with synovial fluid eosinophilia who had Lyme disease. And with the attending who was supervising me during that rotation, we wrote up that case. And I was fascinated by the idea that in synovial fluid, one could see eosinophils, which are rarely seen in synovial fluid. So I got my start in rheumatology writing about synovial fluid eosinophilia, and it happened to be Lyme disease, and that piqued my interest. But subsequently, I've focused more on other forms of inflammatory arthritis, like rheumatoid arthritis, psoriatic arthritis, and axial spondyloarthritis.
Those are conditions that I think have been revolutionized by our biologic treatments. What are these biologics?
Well, biologics for rheumatoid arthritis, the first were approved in 1999, TNF inhibitors, the fusion protein etanercept and the anti-TNF antibody infliximab. Subsequently, adalimumab, another humanized anti-TNF antibody, was released, and then certolizumab pegol, a Fab fragment pegylated of an anti-TNF antibody antigen combining region, light chain, heavy chain, and then finally golimumab, fully human monoclonal anti-TNF antibody. Subsequently, we had abatacept, which is a fusion protein of CTLA4 and the constant region of the immunoglobulin molecule, which is a T cell co-stimulation inhibitor. We have two antibodies against the interleukin 6 receptor, sarilumab and tocilizumab. And then there's, of course, rituximab, which is an antibody to CD20 that depletes B cells. So these are the biologics that are available for rheumatoid arthritis. In addition, for psoriatic arthritis, we've got the anti-IL-17, secukinumab and ixekizumab. And then there's guselkumab, an anti-IL-23, ustekinumab, an antibody against the shared chain that's against IL-12 and IL-23. And I'm probably forgetting some others, but we now have a wealth of biologics that are effective for inflammatory arthritis. We're now a quarter of a century since the first biologics were introduced for the treatment of rheumatoid arthritis.
I remember going to my first advisory board meeting about biosimilars some 10, 15 years ago. And it was with a group of dermatologists, probably who aren't as aware of biosimilars at the time as maybe people in rheumatology. They started by going around the room asking these experts in dermatology what they thought of biosimilars. And each one of them said basically the same thing as they went around the table. They're not the same as the innovator. You can't make them the same as this reference product. So, I just don't trust that it's the same. I mean, unless they'll do 2 phase 3 large controlled trials like the innovator does, I don't trust them. What would you tell people like that?
Well, that was a very common sentiment initially. It's certainly changed now as people have more familiarity with using biosimilars.
Yeah, I think that when people first hear that these drugs are so large and so complicated with tertiary structure, quaternary folding, and aggregates, and they think, it's not the biosimilar is not the same because nobody can duplicate these products. It's kind of scary. But then, if you really understand the principle that nobody can duplicate them, not even the innovator company, you realize that there's batch to batch variation that we totally accept day to day. And the biosimilars basically, I think I've said this to you before, that's basically like another batch of the innovator, and you corrected me because we have more data on the biosimilar than we do for the current batch of the innovator product.
Well, antibody, IgG, has a molecular weight of about 150,000 Daltons, and it's certainly a lot bigger than an aspirin molecule and acetyl salicylic acid. So these are complex proteins that are made by living cells. They're glycosylated and have other post-translational modifications. So it's very difficult, if not impossible, to exactly reproduce a large protein to the point of it being identical to that which is produced by the reference cell line. But biosimilars have been developed as copies of these biologic molecules that are highly similar without clinically meaningful differences. They are studied extensively in over 120 different analytical studies and shown to have no clinically meaningful differences. They're then compared to the reference product in a pharmacokinetic study and shown to be handled by the body the same way as the reference product. And finally, in at least one clinical trial that's really a bioassay in that indication for which the reference product is approved and which is most sensitive to detecting potential differences in efficacy between the reference product and the biosimilar. If the biosimilar successfully demonstrates equivalence in efficacy and pharmacokinetics and high similarity in terms of safety and immunogenicity, the FDA or the EMA reviews the data. And if these criteria are met, then it is approved as a biosimilar, which can be used the same way as another batch of the reference product.
I think people are concerned that biosimilars might be more immunogenic, but my general impression is that as time goes by, companies get better at making less immunogenic drugs, and that on average, the biosimilars may be even less immunogenic than the innovator products.
Absolutely. And in fact, the innovator product might lose efficacy over time. There is batch to batch variation. And for Herceptin, which is a drug used to treat breast cancer, the reference product had a drift in terms of glycosylation, which affected its binding to the Fc gamma R3 receptor, which actually led to less survival. There were more patients treated with more recent batches of Herceptin who were dying from breast cancer than patients who were treated with earlier versions. And the only way that was determined was when it was compared head to head with a biosimilar. The biosimilar had been manufactured in comparison to earlier lots of the reference product and turned out to be more effective or had greater survival than the reference product when they compared it to more recent versions of the reference product. But when they compared the biosimilar to the earlier versions, survival was similar. Reference products drift over time, and sometimes that drift can result in a loss of efficacy. So when a patient with psoriasis stops responding to a given biologic medication, it may be that there is a drift in the manufacture of that medication leading to decreased efficacy rather than an increase in disease activity, which is just not adequately controlled by the biologic medication that had been administered previously.
I'm wondering if there are any proposed regulations you heard of that you thought were just particularly absurd, knowing about batch to batch variation in the innovator product.
I think the regulations for biosimilars are very reasonable. Certainly, they've been more rigorous early on because regulators wanted to be sure that biosimilars were manufactured according to a high standard and that they were compared rigorously to the reference product before approving them. But one of the issues that seems to be going away is the requirement for switching studies to demonstrate that one could substitute a biosimilar for the reference product without the approval of the prescribing healthcare provider, so-called interchangeability. Now, early on, there was a lot of concern about what's going to happen if a patient is on a reference product and then is switched to the biosimilar, and then goes back on the reference product, back on the biosimilar, and so on. Or what happens when you switch among different biosimilars if more than one biosimilar of a reference product is available. But in clinical practice, that's turned out to be a non-issue. In Europe, where there's a lot of experience with biosimilars, and biosimilars have drastically reduced the cost of medication, there's no concern among providers or patients about patients switching among different biosimilars, no increase in loss of efficacy with these switches because the biosimilar is essentially like another batch of the reference product. The FDA seems to be backing away from the requirement for switching studies, which are expensive and take away from the advantage of biosimilars, which is that if there's a lower development cost that's allowed by allowing extrapolation of indications, meaning that if a biosimilar is approved as a biosimilar, it can be used in all of the indications for which the reference product is approved and where the reference product is no longer protected by patent. So now, with the acceptability of changing between a reference product and a biosimilar, the FDA is leaning towards approving biosimilars as being interchangeable at the time that they're approved as being biosimilar, rather than having it be a two-step process. First, the approval as a biosimilar, and then after a switching study shows no loss of efficacy with repeated switching, it then gets a designation of interchangeability. That's probably one regulation that is not necessary, but I wouldn't say that it was a foolish or absurd requirement initially.
Yeah, I think it comes down so much to nobody can duplicate these things, not even the innovator company. And I don't think anybody was concerned about switching from one batch of the innovator to the other batch. And early on, our National Psoriasis Foundation recommended regulations, none of which made sense. Like, you need to inform the doctor when you switch the patient from the from the innovator to a biosimilar. I'm like, well, you're not informing me when you switch from one batch of the innovator to a different batch. So why do you need to have a regulation like that?
You don't absolutely need to inform healthcare provider when the patient is switched, but since the provider is going to have to write for refills of that prescription, it's a good idea with any medication for the provider to be aware of exactly what their patient is taking. Doesn't make a difference in terms of therapeutic efficacy, but it's good to keep records.
This is getting us into some of the practical issues. I've looked at adherence to self-administered biologic medication, and there's so much variability in how well people actually take the medicine that I think it probably swamps any tiny variation there might be between a biosimilar and the innovator or between different batches of the innovator products. One of the things, though, is a real brass tacks issue is these different companies might have different support programs. And if you're switching from one drug to another, it could create huge hassles for an office trying to manage what support programs patients are on. Has that been an issue for you?
It hasn't really been an issue for me. I don't make much use of these support programs. Our nursing staff instructs patients on self-administration, but most patients who used an auto-injector are able to figure out how to use a different auto-injector. The potential advantage of a biosimilar is that it might have a better or easier to use auto-injector So that's a potential way in which a biosimilar can have an advantage for the patient. I don't think it's so much of a hassle. I think the bigger hassle is having to get prior authorization again, and the way in which insurance companies change their formularies. There was one notification that I got that a patient who was doing very well for years on the reference for adalimumab, now their insurance was requiring them to switch to upadacitinib, a completely different class of medication. I would understand if they were asking them to switch to the adalimumab biosimilar, but to force them to switch where there's not necessarily the expectation that it's going to be as effective or as well tolerated, that surprises me to some extent.
I review a lot of papers, and they would, and almost every paper related to biosimilars would say, these are going to reduce costs and improve access. And I'm like, I don't really know because access to biologics has been very good because the support programs, the copay support programs, things like that from companies has been extraordinarily generous. Would be concerned that when biosimilars come, yes, there might be some reduction in cost to the health system, but my individual patient, I can't imagine they're going to see much, if any, improvement in their costs, and possibly it could be worse if the copay support program from the biosimilar that their insurer covers isn't as generous as the one from the innovator company. Are those concerns I should be worried about?
Well, the first point is that Medicare does not allow copay assistance cards to be used. So patients who are on Medicare have to pay a 20% copayment in most cases for their medication. If the medication costs $3,000 a syringe, 20% of $3,000 is $600. So that's quite a significant cost. Many of the biosimilar companies have similar copayment forgiveness cards. Certainly, as a patient taking an expensive biologic medication, it's great to have that copayment forgiveness card, but ultimately, it would be even better if the cost of medications could be reduced for everybody, society as well as individual patients. And the whole purpose of the introduction of biosimilars was to introduce price competition to drive the cost of expensive biologic medications downwards. That didn't really occur too much until there were more than one biosimilar for each reference product. It happened a little bit more when there were two. With infliximab now, with several biosimilars available, three biosimilars that are on the market in the United States, the cost has been reduced by about 80% since the introduction of the first biosimilar. But you really see it with the adalimumab biosimilars, where some of them are discounted 85% lower than the list price of the reference product.
John, are there any other key points we should leave our listeners with?
In the United Kingdom, where there's a single payer system, the National Health Service recognizes that the expense of the preventive therapy, for example, in rheumatoid arthritis, anti-TNF antibodies, offsets the expense of joint replacement surgery. Whereas in the United States, where patients go from one insurance provider to another, the insurance provider that is paying for the anti-TNF therapy has a pretty good idea that they're probably not going to be liable for the joint replacement surgery if the patient develops erosions. And as a result, they're not as likely to pay for the anti-TNF therapy because it's not going to be financially advantageous to them. It's important that the healthcare system recognizes the value of preventive care so that patients are treated most appropriately and do not develop the morbidity and mortality of inadequately treated disease.
Super, John. Thank you so much. I think if our listeners would go and take a look at any of the graphs that show how similar the biosimilar binding is to its target compared to the innovator, and how the pharmacokinetics of the drug in the circulation is, the curves are like totally identical. It's extraordinarily reassuring. So hopefully people feel comfortable with this concept of biosimilars. Hopefully you feel comfortable explaining it to your patients. I want to thank you all for joining us today. This is Dr. Steve Feldman for In Discussion.
Thanks for listening to Medscape's In Discussion Psoriasis podcast series with our host, Dr. Stephen R. Feldman. Be sure to look for more In Discussion episodes wherever you get your podcasts. Check out medscape.com or the Medscape app for show notes, links, and more information on psoriasis.